Pro-life talk. Real world answers. This is Life Report.
Welcome to Life Report. I'm your host, Josh Brom, and I'm here with a really special guest today, Dr. Michael Buratovich, who is the Professor of Biochemistry at Spring Arbor University, and he's the faculty advisor for the SAU Students for Life Club. And he's recently published what is now my favorite book on stem cell research. It's called The Stem Cell Epistles, Letters to My Students About Bioethics, Embryos, Stem Cells, and Fertility Treatments. And in this book, Dr. Buratovich is answering questions from his students and publishing letters that he wrote to his students talking about stem cell research, the humanity of the early human embryo, cloning, fertility treatments, and the viability and future of stem cell-based treatments. And he also blogs at beyondthedish.wordpress.com. And he's a fan of Life Report, which is the most important thing that I've said so far. I don't even know how did that even happen, Dr. Buratovich?
Well, you search for good things on the web, you're bound to run into them occasionally.
So tell me, how did this book come along? This book is awesome. How did this end up happening?
Well, I really have to thank my students for that. My students had lots of questions about early human development, about stem cells, about cloning, and because I studied developmental biology for my graduate degrees, I always had an opinion about this issue. So students would write me letters in the forms of emails. Mm-hmm. I would send them these short emails back. And then I started to say to myself, I really think I need to give them more substantial answers. So I would hold their emails and I'd write these letters, and then I'd hold them and then cross words out and so on and so forth. And pretty soon the word got around that I was kind of the go-to guy on campus for answers to stem cell questions. And eventually, some students actually encouraged me, saying, you should write something about this. So the first iteration of the book was a chapter book. And I took some students across the street to the pizza parlor, and we ate pizza. And I said, guys, what do you think of the book? And they looked at each other, and they looked at me, and they looked at each other, and they looked at me, and they said, it was awful. It was boring. We wouldn't buy this if we were on our last dollar. So I said, well, all right, great. I'm glad you're honest with me. And I really was. I was glad they were honest with me. Right. So I asked them, what can I do to make this more readable? What can I do to make it so you would read it? And they said something that went like this. Write it as though you're talking to us right now. As though you're on a park bench telling us about what you think about stem cell research and cloning and regenerative medicine and so on and so forth. And then something clicked in my head. Hey, what about all these letters they've written me? I can make each chapter a letter with my answers to their questions. And thus was born the Stem Cell Epistles.
Wow.
So I really have my students to thank. And it's not just students, it's also colleagues here at SAU, parents actually. There's some letters from parents, staff actually asked me some questions as well. So I have much to be thankful for with my colleagues and students and so on and so forth.
The book is really conversational, and that actually made it easier or more fun to read. It almost felt like ours being allowed to kind of sit in on almost this private exchange, but a private exchange with someone who's really intelligent and understands the issues. But it was different than just I agree. It was something. It was kind of cooler than just reading. If anyone had just written about, well, here's what stem cell research is. Here's what more like the speeches I've given in the past on stem cell research. Like, this is more fun. There's something really organic about this. So I definitely get that. We haven't done a Life Report episode on stem cell research in a long time. So just to catch up, we've got people that listen on Christian radio and Catholic TV, and just to get everyone on the same page, can you just explain briefly to our listeners what a stem cell is and why they are sought in for research for regenerative medicine?
Well, stem cells have a property that most other cells don't have. They are, arrested might be too strong a word, but they're more primal in terms of their development. They're not as developed as, say, terminally developed cells, say in your skin or in your intestine or your brain or wherever. Mm-hmm. So they have this primal nature in terms of their development. They have the capacity to divide and divide essentially, at least theoretically, indefinitely. They can renew themselves. They have the ability to self-renew. So a stem cell divides, the progeny cell will go on to become something else, depending on what the cell is. The other cell, the parent cell, will stay a stem cell. So there are components in the cell that get divvied up differently when the cells divide. Stem cells also have the ability to respond to a need in the body. If the body is injured or if a tissue is damaged, the stem cell will divide in order to provide cells to replace what's been damaged and to heal the tissues. Stem cells also, in many cases, will actually release molecules that are healing in their properties as well. The capacity the stem cells have to differentiate into one type of cell type or another varies tremendously from one type of stem cell to another. So in the bone marrow, for example, you have a stem cell that is used in bone marrow transplants that can make all of the different types of cells that we find in blood: white blood cells, red blood cells, platelets, the cells that make platelets, megakaryocytes, things like that. Those cells are unique in that they can only make a certain number of cell types, and these are called multipotent stem cells. And there are many examples of multipotent stem cells. Some stem cells can make any adult cell type. We call those types of stem cells pluripotent stem cells. And if a stem cell has the capacity to make every cell type, either of the adult body or the embryonic body,
Right.
then that is a totipotent stem cell. So totipotent stem cells would be the cells of the very early mammalian embryo, the cells of the inner cell mass of, say, a 4 to 5 day old human embryo. Those cells would be pluripotent because they can make all the cell types of the adult body, but not the placenta. Those are pluripotent. And then stem cells in your bone marrow, in your fat, in your skin, in liver, in skeletal muscle, those would be multipotent. And then occasionally you get stem cells that can only make one particular type of cell type. Those would be unipotent stem cells. So that's kind of a brief introduction to stem cells.
Connect that to regenerative medicine. What is it that the medical community wants to do with stem cells?
The medical community would like to isolate these stem cells from your body, expand them, grow them in a dish, and then put them back into your body. And in doing so, provide your body with the raw material to be able to heal what has ever gone wrong with your body. That's really the goal of regenerative medicine. Many different types of cells that you extract from the body don't grow so well when you stick them in culture. It takes a great deal of work to find ways to grow these cells. A good example is the hematopoietic stem cell that we talked about already. The hematopoietic stem cell that in your bone marrow that makes all the blood cells, that cell has been very, very difficult to cultivate. Only really recently, in the last decade, have people figured out ways to expand that particular cell in culture. Other types of stem cells grow more readily in culture, but many of the stem cells from our bodies, as they grow in culture, they'll tend to peter out after a certain time in culture. So trying to find a way to get these cells to grow longer term so they can be expanded to greater numbers. Because remember, Josh, regenerative treatments usually take a large number of cells.
Right.
You have to find a way to be able to grow these cells somehow, to expand them in culture artificially. Now, there's a caveat here. When you grow these cells in culture, you can introduce alterations in those cells. And sometimes these alterations are very bad for the cells. Cells will acquire mutations sometimes, and those mutations sometimes can either cause the cells to die or can transform them into a tumorous type of cell. So you have to also be able to screen cells to determine if they are safe for use in a patient, as well as grow them. So that provides another layer of complexity to this issue. Furthermore, when you put cells into the body, you need to get them to go to the right place. You need to somehow tell them, oh, you're going to become cartilage, or oh, you're going to become bone, you're going to become blood vessel. You have to somehow give them these signals. And one of the best ways to give them these signals is to actually provide some sort of physical scaffold. And various scientists have built physical scaffolds using organic molecules that are easily biodegradable.
Right.
And this is the whole field of tissue engineering. There's a whole journal now of tissue engineering. In fact, there's several journals now of tissue engineering. And there are departments of tissue engineering you can go into as a graduate degree. And if any of your listeners want to be cell biologists, tissue engineering is definitely one of the up-and-coming fields in biology. Trying to make scaffolds from molecules that will be biodegradable, that can be molded and fitted into a bladder, a lung, a kidney, or whatever,
Right.
and then seeding that with stem cells that will find their way into that scaffold, and then embedding into that scaffold growth factors that will cause these stem cells to become certain types of kidneys, blood vessels, blah, 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 so on and so forth.
Yeah.
So these are just some of the goals that scientists have, and it's a very exciting field. Every month you pick up a journal and something really astounding has come across your desk. People are doing really remarkable things with regenerative medicine today.
Now, pro-lifers aren't against all types of stem cell research, so could you briefly distinguish between embryonic stem cell research, adult stem cell research, and also talk about, a lot of people know about those two, not a lot of people know about induced pluripotent stem cell research, so can you just give us a little kind of 101 on those distinctions?
Sure. So embryonic stem cells come from human embryos. You take a 5-day human embryo, you have to somehow eliminate the outer cells. Those outer cells form collectively a structure known as a trophectoderm. You can either use a laser to bore a hole in it, or you can use antibodies that will lyse those cells, destroy them, and then you take those cells of the inner cell mass. Some books call it the embryoblast. It's a bit of a 50-cent word, but ICM, that's, if you say ICM, most people know what you mean. So I'll just say ICM because that's not so 50-cent-ish. You take those cells and you culture them in a very special medium, and if they grow, and that's an if, it's not a guarantee, you can derive an embryonic stem cell line. And that embryonic stem cell line now gives you embryonic stem cells for doing experiments, and the hope was, at least in the past, of using these cells for therapies. There's no way to sugarcoat it. If you isolate embryonic stem cells, you destroy the embryo. There's just really no way to sugarcoat it. Now, there was a procedure that was published several years ago by Tony Lanza's lab, where he took very, very young embryos that were still at the 8-cell stage, 16, 12-cell stage, and he isolated individual cells from those, and he was able to take those and grow them in culture. And these cells had all the characteristics of human embryonic stem cell lines, and because procedures like this are done for pre-implantation genetic diagnosis, people in the field of stem cells and also policymakers have said this is a way to make human embryonic stem cell cultures, in fact, personalized embryonic stem cell cultures, without destroying the embryo.
Right.
The problem is that in order to justify this, these researchers have said, well, we have kids, over 1,000 kids who've been born who, when they were embryos, and it's interesting, they acknowledge the continuity between those two as embryos and now as toddlers or preteens or something like that, and they're normal. What these studies do not address is what happens to these children later on, because mouse studies have shown that you can do the same procedure to mouse embryos. You'll get normal mice if you then take these embryos and implant them into a surrogate mother. The mice will be normal, but later on they will show profound neurological problems. So we might be subjecting these individuals to very, very severe risks, and frankly, without their permission.
Right.
Would we subject a toddler to this type of risk? As a father, and as a father myself, my answer would be hands down, no.
Right.
And yet we're saying, oh, we can do this. So they can have their own toolkit? Well, wait a minute. What if we don't need a toolkit like a human embryonic stem cell line? What if there are other stem cells we can use? And in fact, you can take adult cells, and through genetic engineering techniques, you can overexpress particular genes in them, genes that are actually expressed in embryonic stem cells. And these cells will then de-differentiate into a cell that's very much like an embryonic stem cell. Now, let me be clear here. These cells are not exactly like embryonic stem cells. They're very, very similar. They express many of the same genes, many of the same proteins, Culture-wise, they have many of the same culture features,
Mm-hmm.
at least under the microscope, that you see in embryonic stem cells, but they're not exactly the same. So people say they're embryonic stem cell-like, and I think that's a fair description. But these particular stem cells, one, they're personalized. They have the same barcodes on the outside of their cells that your body has.
Right.
Secondly, no embryos need to be destroyed in order to make these cells. Now, let me just add, when these cells were first made, they were made, Shinya Yamanaka won the Nobel Prize 2 years ago in medicine. He shared the prize with John Gruden for his discovery, his invention really, of induced pluripotent stem cells. So this is a remarkable find.
Yeah.
But he made them using a particular group of viruses, retroviruses, that insert into the genome of the host cell.
Mm-hmm.
They cause mutations. There are ways now to make these without using any sort of tools that insert into the genome of the host cell. But then there was a concern that this whole reprogramming process tends to cause mutations. However, those papers, which were published in 2011, overestimated the mutation rate. And other papers that were published later 2011, 2012 have shown, if you sequence the whole genome of induced pluripotent stem cells, and this is something to keep in mind, the quality of an induced pluripotent stem cell is very cell line specific.
Mm-hmm.
Some of these cells will have lots of mutations and will cause tumors and will be potentially dangerous to use. Others are not. So you have to take these cells cell line by cell line, and you have to screen them carefully. And the screening techniques are getting better and better now.
Interesting.
And people, in fact, in Japan, there is, in fact, you can find this on my blog site, there are a couple of clinical trials that are online pretty soon. One is to treat people with particular types of blindness, and the other is to actually make platelets, because platelets don't have nuclei. So you can make the cells that will make platelets, megakaryocytes, and then put those into the bone marrow, and then they'll make platelets for people who have these platelet disorders. These are clinical trials that should be in the works, I would think, by next year easily.
So a lot of people argue that, look, these embryos are so small, they're just a tiny clump of cells, they have no nervous system, no heart, no consciousness, no ability to feel pain, like all these things that a lot of people think are really important and maybe even a necessary condition for being a valuable person. And they think it's just crazy for people like me who think that that embryo, even at 5 days old, is a valuable human being that's intrinsically valuable, that we should not kill for their stem cells. So what do you say to those people that just like they look at the ICM and they're just like, really? That's what you're trying to protect?
I think that objection has some intuitive appeal, because as you look at the embryo, it really doesn't look like us. It's pretty hard to get worked up about it. And I had this conversation with Scott Klusendorf.
Mm-hmm.
Scott is for the use of images of abortion in order to show people what abortion actually is.
Mm-hmm.
And I said, Scott, if you do that with embryos, people are not going to get all that torn up about it.
Right.
The simple fact of the matter is that I think we have to agree, yes, the embryo, as far as we know, is not conscious. The embryo does not have a nervous system, does not have a circulatory system, does not have skeletal muscles, but we have to qualify that because it doesn't have those things yet. The fact is that the embryo looks exactly the way it should look at this point in development.
Some people think that we should use cloned embryos to solve the practical issues with embryonic stem cell research. If you clone embryos, then you're probably not going to have the immune system problems because they came from your own body. They've got the same blueprint. Do you believe that human cloning could be an ethical alternative?
I do not, no. Cloning of human embryos was a big, big technical problem that was actually solved just a few months ago by a gentleman named Matilipov at the Oregon University of Health Science. And the way he did it was remarkable, the way he solved the problem. But he was able to use cloned human embryos to make human embryonic stem cells from those cloned embryos. The simple fact of the matter is that when he examined the gene expression patterns from those cloned embryos, they were virtually indistinguishable from embryos made from in vitro fertilization. Those embryos were made only to be destroyed. And pro-life advocates have called this clone and kill, And that's exactly what it is.
Right.
Even if you look at mouse embryos and look at where mouse embryos start to go wrong, mouse embryos, if they have problems, they usually have problems after implantation. The early stages of embryonic development are still normal. The fact that human embryos that are produced by the process of cloning essentially have an appearance and a pattern of expression of proteins and genes and so on and so forth that are essentially normal. These are human beings.
Right.
They were not made by virtue of fertilization, but think of an identical twin. An identical twin that results from the splitting, essentially, of a very early embryo. That identical twin wasn't made by virtue of fertilization either.
Mm-hmm.
So how the embryo began is really not all that significant. It's what it is. What it is, it's a product of human entities, an egg and a somatic cell. These are analogous to an egg and sperm.
Right.
This biological process produces a human individual, a very, very young human individual. Cloning, I think, is just as immoral as embryonic stem cell research.
Okay, so probably I think the argument that I've heard the most often in response to pro-lifers that are concerned about embryonic stem cell research is, look, it's kind of a utilitarian argument. There's already all of these embryos that are already frozen. They're leftovers from in vitro fertilization. Okay, maybe we won't create new embryos just for this, because maybe some people can even understand why we'd be concerned with that. But look, we've already got these leftover embryos from IVF. Why not put them to some good use instead of them just getting thrown away, discarded, or just lying in some cold storage unit for years and years and years? How would you respond to this kind of more utilitarian argument?
I think that argument has tremendous intuitive appeal. And the problem is largely due to the very unwise regulatory framework we have for in vitro fertilization in this country. The simple fact of the matter is that this argument, I think, only works if you already assume that very young human embryos are not human persons.
Mm-hmm.
If you think of any other age of humanity, toddlers, neonates, adults, and if we say, look, you're going to die anyway, this argument simply would not work. Josh, all of us are going to die at some point in time. If we were to push that argument, then really you could justify killing anyone on the basis of that argument. And Scott Klusendorf gives, I think, a remarkable analogy that I use in my book. I must give Scott the credit for this, and that is, if you were on an island and you had an orphanage full of orphan children,
mm-hmm.
which is very much what these embryos sitting in cold storage are. And we really don't know how many embryos we have in cold storage. The Rand Corporation did a study that said it was on the order of about 300 to 400,000. It's probably over 500,000 by now. It may even be upwards close to 1 million. We don't know. And it was very clear that food and water and other types of materials were simply not going to be available to you. So you just went to the kids and said, here's what's going to happen. The whole, almost like a George Mueller type of situation.
Mm-hmm.
Here's what we're going to do. We're just going to do the best we can with what we have. And a physician came to you and said, look, I will take these kids, I will take care of them, but they are going to be used for medical experiments that will be used to cure thousands of people. The kids will have to be sacrificed in the midst of the experiments. The kids will, in fact, will be dismembered as a result of these experiments, but they're going to die anyway.
Right.
I don't think you could find a single person alive who would say that the head of that orphanage would do a virtuous thing if he or she then turned those kids over to that physician. You have to essentially assume the embryo is a non-person in order to make this argument work. And I think really the only humane thing to do is to implant them. I know that sounds perhaps unworkable. It does sound unworkable, but these embryos were made. They are sitting in cold storage, and that's the obligation, moral obligation I think we have by making them. And I think this moral obligation has really left us because of this unwise model that governs in vitro fertilization in this country.
What should pro-lifers be doing about stem cell research? There's a lot of pro-lifers that are talking about abortion, but there's not, I don't think, a lot of people that are talking very much about stem cell research and human cloning. What would, if you could have them do any one or two or a few things, what is it you would have them doing besides following your great blog?
Why, thank you. Number one, I think the reporting on the stem cell issue is egregious. If you go to my blog site, I point out some particular howlers that come from the press. I think if you see bad writing about the stem cell issue, I think, first of all, pro-lifers should be educating people about the stem cell issue. Secondly, I think they should connect the stem cell issue to the abortion issue, because part of the reason why the stem cell issue, I think, is not going to go away is because if the scientific community gives away the stem cell issue, they've given away the abortion issue.
Right.
And I really do think that the abortion issue drives this to some extent. So pro-lifers should connect the stem cell issue to the abortion issue, because life is a continuity and it's an unbroken continuity. Thirdly, I think they should be very educated about the facts of human development, especially early development, and also the facts of stem cell research. And I think they should use that. They should bring that up constantly. And again, make this connection, make it deliberately. And fourth, I think they should write their congressperson about why their tax dollars should not go to money should not go to laboratories that are using it to make new embryonic stem cell lines. We don't need them now that we have induced pluripotent stem cells. We have enough of these stem cell lines now that are available to use as standards. We really don't need to be killing more embryos to make these.
It's like we've got these principled arguments. We've got reasons to believe that human embryos are just as valuable as all of the other human beings that are older, but we also have these pragmatic arguments as well, that we have other things going. I think if you put those both together, they make a very powerful chord. Dr. Michael Buratovich is a professor of biochemistry at Spring Arbor University. His book is The Stem Cell Epistles, Letters to My Students About Bioethics, Embryos, Stem Cells, and Fertility Treatments. Oh, we're both holding it up. Okay, I think everyone will get it now. He blogs at beyondthedisc.wordpress.com. Dr. Buratovich, thank you so much for joining us today. That is our show, and remember, you can have a great impact on any conversation if you remember to ask good questions, listen to understand, and find genuine common ground when possible. Now go talk to somebody.
You've been watching Life Report, pro-life talk, real-world answers. Life Report is produced by Right to Life of Central California. Visit their website at fresnoprolife.org.